Well, I'll tell you one thing, Clyde. This is chapter 16 from the textbook by Saladin, and we could take an entire semester dealing only with this particular topic. Well, we don't have the entire semester. We've only got a couple of days to get you ready for your final exam. So I will summarize things and be as brief as I can. All the same, I'm looking here and there's 21 slides that deal with the senses only. So I will try to make it as concise and as brief as I can. All right, the senses, we have six of them. I'm not sure where the movie came from called The Sixth Sense. We already have six, and here's a kid claiming to have a sixth sense that he sees dead people. Well, I don't know about that. We already have six, and they are the sense of vision, hearing, smell, taste, equilibrium, and touch. Let's move along. Well, we are still talking about neurons and the special property that neurons have, the ability to fire an action potential. Well, an action potential has to begin somewhere. So in the sensory pathways, we begin at what is called a receptor. Receptors are biological entities that will fire an action potential in response to certain stimuli, such as physical pressure. Those are called mechanoreceptors. Obviously, we have mechanoreceptors in the skin, our sense of touch. We also have mechanoreceptors in the inner ear. Our sense of hearing and our sense of equilibrium have mechanoreceptors which initiate the firing of an action potential. Thermoreceptors are sensitive to hot and cold. Obviously, those would be present in the skin. Nociceptors are stimulated by pain. Pain oftentimes is mediated by chemicals released by injured cells, such as the chemical bradykinin. They will bind to nociceptors, and the nociceptors initiate an action potential. Chemoreceptors, such as our sense of smell and sense of taste, those receptors are stimulated by the binding of certain chemical compounds. And then last but not least, perhaps most impressively, our photoreceptors. These are the rods and cones of the retina. They are stimulated to fire an action potential anytime they are impacted upon by certain wavelengths of light. Well, let's begin our tour of the sensory organs and tissues by looking at our sense of vision. So our eyeballs have to move. They move in response to the contraction of muscles. We have four muscles that attach to the eyeball and pull the eye in a straight line. Well, the word rectus means straight. So we have the superior rectus, which pulls the eye straight up. We have the lateral rectus, straight to the lateral aspect or to the side. The medial rectus moves the eyeball toward the nose. Inferior rectus moves the eyeball straight down. So those are four muscles that move the eyeball in a straight plane. Then we have two, the superior oblique, which will move the eyeball down and out. The inferior oblique moves the eyeball up and out. So oblique means at an angle, rectus means straight. So let's talk about innervations. All of these muscles are innervated by cranial nerve number 3, the oculomotor, except the superior oblique, which is innervated by cranial nerve number 4, the trochlear nerve, and the lateral rectus, which is innervated by cranial nerve number 6, the abducens nerve. Remember, to abduct means to move toward the side, so that's why the nerve is called abducens. The lateral rectus moves the eyeball toward the sides. Well, in the image on the left, I think the point has been made before. I'm not sure where I made it. The eyeballs are exposed to the outside. The outside is poisonous to our living cells, so we have to protect our living cells from contact Otherwise, our eyes would chronically be inflamed. So we have there the lacrimal gland, innervated by parasympathetic fibers from the facial nerve. We release tears onto the surface of the eyeball every time we blink. Those tears are forced into the nasolacrimal duct where they drain into the nose. And if they're not lost to the outside, then they are swallowed, and whatever microbes or crap is on the surface of the eye gets washed into effectively the stomach and its nice little hot tub of hydrochloric acid. On the right side, we have a pretty detailed look at the orbit and the extraocular muscles. Notice there we have the levator palpebrae superioris, which you have already encountered. That raises the eyelid. The innervation there is cranial nerve number 3, oculomotor. I went through the other extraocular muscles on the previous slide, so I won't do that again. They are shown there. Notice that the cornea is the same layer of tissue as the sclera. The sclera is the outermost tissue of the eyeball. It is white. So when you see the sclera, you are looking at the same layer of tissue that becomes the cornea. The only difference between the two is that the cornea is clear. So we have to allow light to pass through the cornea so it is clear. Otherwise, it is continuous with and the same as the tissue which we call the sclera, the white of the eye. The word corny and the word sclera both mean hard. So they are the same tissue. Cornea means hard, sclera means hard. We also sometimes throw in another prefix, kerato, which also means hard. So an infection of the cornea is called keratitis. A malformation of the cornea is called keratoconus. Well, kerato means corny, which also means sclera. All of those mean hard. Here we have a mid-sagittal view of an eyeball, and what I want to do here is follow the path of light as it enters into the eye and as it impacts upon our photoreceptors. So light coming in must pass through the cornea. The cornea is clear, but even so, it is not air. Anything that is not a vacuum will bend light. The cornea is no exception to that. Light is bent as it passes through the cornea. If the cornea is not shaped properly, then the bending of light will cause blurry vision. That is called astigmatism. I suspect some of you have corneas that are not shaped perfectly. Consequently, you have the blurry vision that we call astigmatism. Once we pass through the cornea, we're in the anterior chamber. The anterior chamber is filled with a fluid called aqueous humor. It is the aqueous humor in the anterior chamber that can cause the condition called glaucoma. Anything that is produced 24/7 has to be absorbed 24/7. Otherwise, it's going to increase in volume and therefore increase in pressure. So if we have a production of aqueous humor in the anterior chamber and it is not equally drained by the canal of Schlemm, then the pressure is going to build up in the anterior chamber. That pressure eventually will translate back to the retina and will cause blindness if it is not corrected. Glaucoma is the second leading cause of blindness in America. Okay, from the anterior chamber, we have a hole that passes through the donut-shaped iris. The iris is the pigmented part of the eye. My irises are brown. I don't know about yours. It is a sphincter muscle. It's circular. Well, we call circular muscles sphincters. And if it contracts, well, the pupil dilates. If it relaxes, well, the pupil constricts. the pupil is a hole. A hole is the absence of matter. So what we have then is a way for light to enter through the anterior eye and then go into the posterior chamber, which is also filled with aqueous humor. All right, the light then passes through the lens. The lens is a biconvex structure which has the power to bring rays of light to a focus. So when we pass rays of light through a biconvex lens, then the rays converge or come to a point of focus. That's what we must have in order to have clear vision. Posterior to the lens, that is called the vitreous body. It is filled with a jelly-like substance called vitreous humor. Okay, the light then passes directly through the vitreous body, and if we focus it properly, it comes to a point of convergence at the fovea centralis, which is located there at the retina. So the first layer that we come to is the layer of rods and cones. We call that the retina. External to that is the vascular layer called the choroid, and surrounding that is the outermost layer, the sclera, which is one and the same with the cornea. Okay, we will detail the layers of the retina on the next slide. Right now, just notice that we have the optic nerve, which is simply the continuation of the retina as it goes to the visual cortex in the brain. Okay, when we take an up-close look at the retina itself, well, let's begin right at the very bottom of this illustration. The outermost layer of the eye is the sclera. The layer that is internal to that is the vascular layer. We have blood in the choroid, and then internal to that is our retina. The retina has the photoreceptor cells that we call rods and cones. Cones are sensitive to colors. Rods are sensitive to grayscale. So we have night vision courtesy of the rods. We have day or color vision courtesy of the cones. Now, when Roy G. Biv, red, orange, yellow, green, blue, indigo, and violet, those are the colors of the visible, those are the wavelengths that our retina is able to fire an action potential in response to. So we have Roy G. Biv. Those wavelengths will prompt the rods or cones to fire an action potential. The action potential then goes to the bipolar cells, which then go to the ganglion cells. Now, also pictured there are the amacrine cells and the horizontal cells, but they are not in the visual pathway. They are feedback cells. The cells that are in the visual pathway are the rods and cones, bipolar cells, ganglion cells. Now notice that the axons of the ganglion cells all come together. They form the optic nerve. So that is the visual pathway to that point. Rods and cones, bipolar cells, ganglion cells, and the axons of the ganglion cells are the optic nerve. So on the next slide, we will take it from there. Okay, so looking at this diagram here, we can complete the visual pathway. So we initiate the sense of vision at the rods and cones. They are the receptors. The ganglion cells issue their axons. Those axons become the optic nerves. The optic nerves cross right in front of the pituitary gland at the optic chiasm. After the crossing, then the continuation of the visual pathway, those are called the optic tracts. Notice that we are going to issue connections to the pretectal nucleus. Also, the superior colliculus is in the visual pathway, and our synapse in the thalamus is at the lateral geniculate nucleus of the thalamus. So coming from the optic tracts, we make connections with the pretectal nucleus, with the superior colliculus, and the lateral geniculate nucleus of the thalamus. The continuation of the pathway from that point is called the optic radiation. The optic radiation will send axons to the visual cortex. The visual cortex is located in the calcarine sulcus of the occipital lobes. That's where we perceive that we have seen something. We then have the visual association areas, which enable us to make sense out of or to identify what we have seen. So if you want to memorize the visual projection pathway, go rods and cones to the bipolar cells, to the ganglion cells. The axons of the ganglion cells are the optic nerves. The optic nerves cross at the optic chiasm. Posterior to that point, we call those the optic tracts. The optic tracts will make connections with the pretectal nucleus, the superior colliculus, and they will synapse in the lateral geniculate nucleus of the thalamus. Remember, the thalamus is the switchboard for incoming sensory signals. So it will direct that action potential or those visual signals to the visual cortex. The visual cortex is in the calcarine sulcus of the occipital lobes. So that is your visual pathway. Well, there are three dimensions to the space around us. I mean, we have the front to back direction, we have the top to bottom direction, and we have the left to right direction. So there are three different directions from which compressed air molecules come at our head. Well, we have positioned on the side of the head the auricles. The auricles function as a funnel, so they are able to catch the waves of compressed air molecules. Those molecules are compressed anytime something moves in space. Now, listen to me. I'm going to clap my hands together. Here I go. Well, you heard that. Well, what I did was compressed air molecules together, and those compressed air molecules travel in waves. Those waves are captured by the auricles, the external projection of our ears on the side of our heads. They function as a funnel. So they're going to take those waves of compressed air molecules and force them into a smaller and smaller space. Well, that's what a funnel does. So the small space that we're going to there is the external acoustic meatus. You remember that? Okay, you can memorize the structures of the external ear. The helix, the antihelix, the tragus, the antitragus, and the lobule. We also have an area called the triangular fossa, and we have an area called the concha. So those are the anatomical structures of the auricle and the function of the auricle. And you say, well, how important could that auricle be? Well, smart people who study ears have said that without an auricle, if your external auricular appendage is cut off, you would lose half of your hearing. The only thing that you would hear on that side is waves of compressed air molecules that are coming directly at your external acoustic meatus. Those are the only things you would hear if you didn't have an auricle on that side. So in order to capture things coming from the front to the back, or from the back to the front, or from top to bottom, you have to have an auricle. You have to have a funnel to direct those things into the ear hole. I mean, the external acoustic meatus. Well, once the auricles have funneled the waves of compressed air molecules into the external acoustic meatus. By the way, the auricle there is labeled pinna. Sometimes it is called the pinna of the ear. I prefer auricle. Anyway, if we take those waves of compressed air molecules and we funnel them into what is called here the auditory canal, then the auditory canal has a dead end. There is a membrane there that is the tympanic membrane or the eardrum. So what we have is energy coming into the auditory canal. The energy is the compressed air molecules traveling in waves. When they strike the tympanic membrane or the eardrum, it causes it to move. Well, notice that the eardrum is attached to the malleus, that's a bone, which is attached to the incus, that's a bone, which is attached to the stapes, that's a bone. So if the eardrum moves, then those three bones also will move because they're all attached. So those are the auditory ossicles, or the three bones of the middle ear. Notice that the stapes is attached to the cochlea. The cochlea and the vestibule are fluid-filled chambers. The stapes is a bone. It attaches to those fluid-filled chambers. If the stapes moves, then that will create waves in that fluid. The fluid inside the cochlea is called endolymph. So anytime waves of compressed air molecules strike the tympanic membrane, it moves. If it moves, then the auditory ossicles move. The third auditory ossicle is the stapes, and it's attached to this fluid-filled chamber. Well, the fluid inside the cochlea will move in waves. Notice that the cochlea is a snail shell shape. It is simply a coiled tube that is filled with fluid. So the fluid will move inside the cochlea. When we take a cross-section of the cochlea and look at its internal anatomy, we'll see that it has three chambers. So on the next slide, I will show a cross-section of the cochlea, and in there is our organ of hearing, which is called the organ of Corti. All right, one other thing before we leave this particular diagram. Notice that the middle ear is attached to the nasopharynx. Well, that is to equalize pressure on either side of the eardrum. So if you go up on a mountaintop or if you go up in an airplane, the air pressure is lower. So if that is the case, that would make the eardrum bulge out and it would hurt. Well, we can equalize that pressure by swallowing or by yawning or etc. Alternatively, if you went 10 feet down in the water, the weight of the water pressing against your eardrum is going to push it inward, and eventually that's going to hurt. So you can hold your nose and push and equalize the pressure to avoid the rupturing of your eardrum. Also, one other thing that you might be wise to concerning the eustachian tube, since there are about 100 species of bacteria that live in the throat, some of them can migrate by way of the eustachian tube from the throat to the middle ear and cause otitis media, or infection of the middle ear, earache. Next slide, please. So if we take the cochlea, which is simply a coiled tube that is filled with fluid, and we cut it and look at it in cross-section, what we see is that the tube has three different fluid-filled chambers. Those are the scala vestibuli, the cochlear duct, which is also called the scala media, and then down below the scala tympani. Well, we have membranes that separate those three fluid-filled chambers. If a sound is made, which is compressed air molecules hammering against the eardrum, creating waves in this fluid, the fluid is inside these chambers, well, it's going to cause the membranes to move. We have then on the basilar membrane what are called hair cells. These are actually epithelial cells that have cilia on the top of them, so that's why they're called hair cells. Over the basilar membrane, we have the tectorial membrane. Remember that tectum means roof. Well, look there at the organ of Corti. Those are the epithelial cells with their little cilia coming off the top. Well, there is a roof over them. The basilar membrane will move up and down like a sailboat goes up and down on the ocean as the waves go by. Well, remember, we're talking about waves in this fluid. So as the waves pass under the basilar membrane goes up and down. The organ of Corti is positioned between the basilar membrane, which moves, and the tectorial membrane, which doesn't move. Well, notice that if the epithelial cells with their little cilia coming off their top move up, then the cilia are going to come into contact with the roof, the tectorial membrane. That will cause the cilia to bend. When the cilia of those hair cells are bent, potassium channels are opened in those cells. That's where the action potential that we call hearing begins. Now, you may be curious about hearing loss. Those cilia are fragile. The tectorial membrane does not move. So if a sound is too loud, it will cause the basilar membrane to come up and compress those little cilia too hard against the tectorial membrane, and they will break. When I was teaching in the laboratory, I would give the story of when I was growing up, my family had the only trampoline in town. In the wintertime, we lived in a big house with high ceilings. We would bring the trampoline inside. Well, those of us who had some experience knew better than to jump too high on an indoor trampoline. But when guests would come by, which was all the time because people loved to come to my house to jump on my trampoline, well, we wouldn't tell them, don't jump too high. We would just let them learn the hard way. So they would get on the trampoline, they'd go to jumping, and wham, they would hit their head on the ceiling. You get the picture here. When the basilar membrane goes up and down, which it does as the waves of endolymph flow under it, the basilar membrane goes up and down like a sailboat on the ocean. Well, the tectorial membrane doesn't move. So the little hairs, the cilia, press against the tectorial membrane. When they are bent, potassium channels are open, which causes the cells to depolarize, and we call that depolarization of the hair cells an action potential. The action potential is then transferred to the fibers of the cochlear nerve. So you can see it there. So that's where we begin our sense of hearing. On the next slide, we'll take up the auditory pathway so you can see where it goes from there. Okay, so begin the auditory pathway then at the organ of Corti, which is located in the cochlea, situated between the basilar membrane and the tectorial membrane, the little hair cells with their cilia. When those cilia are bent as they are compressed under the tectorial membrane, potassium channels open, which depolarizes those cells. That's where the action potential begins. So that is picked up by the cochlear nerve. So that is our first order neuron, cochlear nerve, which goes to the cochlear nucleus of the pons. So that's our first order neuron. We send fibers down to the medulla oblongata, the superior olivary nucleus, but there is no synapse that takes place there. So this is still our our second order neuron. The first order neuron goes from the organ of Corti to the cochlear nucleus, synapse there with the second neuron. The second neuron goes to the inferior colliculus of the midbrain. It does synapse there. That's where our third neuron begins. From the inferior colliculus, we go to the medial geniculate nucleus of the thalamus, synapse there. So we have then a fourth order neuron in the auditory pathway going from the thalamus to the superior temporal gyrus. That's where our auditory cortex is. That's where we perceive that we have heard something. We then have surrounding the auditory cortex the auditory association areas where we make sense out of what we have heard. So the auditory pathway is actually a 4-neuron pathway. Most sensory pathways are 3 neurons. The sense of hearing offers 4 neurons. So it's a fourth order neuron going from the thalamus to the auditory cortex. For the sense of smell, what we have are chemoreceptors. These are receptors that will bind certain chemical compounds, and when those compounds bind, an action potential is fired. We don't have a very keen sense of smell. The number of chemical compounds that we can bind is very few compared to what other animals, such as a dog has a much more keen sense of smell than a person. All the same, the dynamic is the same. We bind chemical compounds that are in the air that we breathe. When that binding takes place, depolarization occurs, action potential is fired. So remember that we have the cribriform plate, and in the cribriform plate of the ethmoid bone, there are little bitty holes that we call the cribriform foramina. Through the cribriform foramina, the olfactory nerves enter into the nasal fossae. So the nasal fossae are open to the outside. So we actually have nervous tissue in contact with the external world. That is very unusual. Something else that's very unusual, we talk about nerve cells being non-replaceable. Not true here. Since the olfactory nerve cells are in contact with the poisonous world, they can be easily lost. Well, strangely enough, they are replaceable. So we do have evidence of some neuron mitosis that takes place in the olfactory sense. Okay, so we then collect together the olfactory nerves. I will detail the connections made there, all coming together to form the olfactory bulb, which sends their projections, their axons, by way of the olfactory tract to the olfactory cortex. I think I have that detailed a little bit better on the next slide. All the same, I wanted to point out to you that this is how the sense of smell is positioned in the nasal fossae. We have chemical binding cells, they're called odorant binding proteins, located on those olfactory nerves, and that's where the depolarization and therefore the action potential begins. Well, this is a very nicely done little diagram to give you a close-up look of how this happens. Those olfactory receptor cells, well, we just call those our olfactory nerves. Those are the ones that are somewhat replaceable because they're exposed to the environment. Notice that the axons pass through the cribriform foramina, the holes in the cribriform plate. We come together to form what are called glomerular cells, and we synapse with the second order neuron in those glomeruli. So the second order neuron there is called a mitral cell. We also have cells located in there called tufted cells. They are not in the olfactory pathway. They actually feed back in the other direction. So where we're going to here, or where we're coming from and going to, is the olfactory receptor cell, that's the first order neuron, to the mitral cell, synapsing in the glomerulus, or the glomerular cells that are located there in the olfactory bulb. From there, the axons of the mitral cell form the olfactory tract. The olfactory tracts will then go to the olfactory cortex, which is in the inferior gyrus of the temporal lobes. Okay, this illustration is a little bit busier than I would like for it to be, but that's okay. I think you can do it. So once we have the axons of the mitral cells exiting from the olfactory bulbs, then we are in the olfactory tracts. We are going directly to the olfactory cortex, which is the very bottom noodle, the inferior gyrus of the temporal lobes. So this is where we perceive that we have smelled something. All right, notice that is kind of an unusual arrangement because usually we synapse at the thalamus, and the thalamus directs sensory signals, not in the case of the sense of smell. We go directly from the olfactory tracts to the olfactory cortex, and then later we will make connections with the thalamus, also the hippocampus. Remember, I told you that our sense of smell is very strongly remembered. Also, I told you that the sense of smell carries with it very strong emotional connotations. Well, those are limbic system structures, the hippocampus, the amygdala, the hypothalamus. So we make connections with the thalamus, but it is only secondary to having passed through the olfactory cortex. So it's a little bit different from the other sensory pathways, but only in that sense. Just remember that we do, with the sense of smell, have a very strong influence upon the limbic system structures, memories, and emotions associated with our sense of smell. Now, notice that we also have down at the very bottom connections to the reticular formation. Well, some smells we don't like, and so we get, they can make you sick. So there's the connection to the reticular formation. Smells that are disgusting and can cause you to vomit by stimulating the emetic nucleus of the medulla oblongata. Emesis means vomiting, and we do have a nucleus in the medulla which will prompt a person to vomit if it's stimulated. So these smell connections actually go there to that emetic nucleus in the reticular formation. Well, if you stand in front of the mirror and stick your tongue out, you can see that the tongue has coming off of its surface all sorts of little projections and bumps and mounds. Those are called papillae. We have four types of papillae. The ones that are most numerous, that kind of give the surface of the tongue the appearance of carpet, those are called filiform papillae. Filiform papillae have no taste buds. They are, however, sensitive to the texture of food. They are innervated by branches of the vagus nerve, cranial nerve number 10, and so we have some appreciation for the texture of food, courtesy of the filiform papillae. A little farther back, larger, shaped like mushrooms, consequently called fungiform papillae, shaped like mushrooms. These do have 2 or 3 taste buds per papillae. So the fungiform papillae do participate in the sense of taste. Not shown here for reasons I don't know, but over to the sides of the tongue, there are papillae called foliate papillae. They have taste buds in a child, but those taste buds usually undergo atresia by the age 2 or 3, so they're not even shown here. Now, the ones making a V shape there going toward the posterior of the tongue, there are usually 8, 9, or 10 of these valate papillae. The valate papillae are rich with taste buds, having about 250 or 300 taste buds per papillae. So those are the papillae that have most of the taste buds. We innervate the tongue by way of the facial nerve. The anterior 2/3 of the tongue is innervated by the facial nerve. The posterior 1/3 of the tongue is innervated by the glossopharyngeal nerve. We pick up on 5 different taste sensations. We have sweet, sour, bitter, salty, and umami. Umami is a meaty taste. It is the perception of amino acids in the food. Umami. Okay, so let's look at the next slide here to get a close-up of what does a taste bud look like and how does that signal get to the gustatory cortex. Okay, in the big picture of the tongue there on the left, it does in fact show the foliate papillae on the left and right side that I mentioned on the previous slide. After age 2 or 3, the foliate papillae don't have any taste buds. The fungiform papillae have a few, the filiform papillae have none. So most of our taste buds are located there in the valate, or what is labeled there, the circumvallate papillae. So looking at a taste bud up close, the taste bud opens to the surface of the tongue. That is called the taste pore. So the tongue has to be wet. If the tongue is dry, then nothing can go down into the taste pore. So the tongue has to be wet in order to have a sense of taste. So whatever is on the surface of the tongue will drip down into the taste pore, where it will come in contact with the taste hairs. They're called here the gustatory hair. The taste hairs or gustatory hairs are projections off of the taste receptor cells. So this is where we bind the chemical compound. The taste hair binds the chemical. When the chemical binds to the taste hair, it causes the opening of potassium channels, which depolarizes the receptor cell. So we have in the taste bud 2 other cell types. Notice the taste receptor or the gustatory receptor cell is colored purple or lavender. Well, we also have some that are colored kind of pink, and down at the bottom we have some round shaped cells. Well, they are also important here. The pink colored cells are simply called supporting cells. And then down at the bottom, those big round shaped cells, they're called basal cells. Well, notice that the taste receptor cells are exposed to the outside. They are epithelial cells, they're not nerve cells. They will have to be replaced. I mean, you get hot things and abrasive things on your tongue, it can destroy those taste cells. Well, they're going to have to be replaced. So we have mitotically active basal cells there at the base of the taste bud to replace the taste cells that are routinely lost. Okay, the innervation from the taste receptor cell, if it is the anterior 2/3 of the tongue, those are branches of cranial nerve number 7, the facial nerve. If it's the posterior 1/3 of the tongue, then those are branches of the glossopharyngeal nerve, cranial nerve number 9. Okay, we will take it from there to the gustatory cortex on the next slide. Okay, so the receptor cell, or the cell that initiates the depolarization and the action potential, those are epithelial cells that are simply called taste cells or gustatory receptor cells located in the taste buds. They are innervated by the facial nerve out front, the glossopharyngeal nerve in the back part of the tongue. So the transmission of the signal goes from the taste receptor cell to the facial or glossopharyngeal nerve. That is our first order neuron. The synapse that takes place in the medulla oblongata, that is called the solitary nucleus. So that's where we begin our second order neuron. The second order neuron goes from the solitary nucleus of the medulla to the thalamus, where it synapses with the third order neuron. The third order neuron goes from the thalamus to the gustatory cortex, which is located in the insula, where we perceive that we have tasted something. Ah, that was some good strawberry shortcake. All right, what you see here is the organ of two different senses. We have already talked about the sense of hearing. That is the job of the cochlea and the receptor inside the cochlea, which we call the organ of Corti. That is a different structure from the vestibule. The vestibule is about our sense of equilibrium. Equilibrium is an awareness of our position in space and our movements. Are we upright? Are we upside down? Are we moving left, moving right, moving up, moving down? Those are vestibular functions. So the individual components of the vestibule include the saccule, a fluid-filled chamber, the utricle, a fluid-filled chamber, and the three semicircular ducts, which are fluid-filled tubes. It is the same fluid shared with the cochlea. So all of this taken together oftentimes is simply called the labyrinth, and disorders of the inner ear are often called labyrinthine disorders. So the same endolymph and the same perilymph that are present in one are present in the other, but the senses are totally different. Hearing and equilibrium are two totally different senses. So what we have then inside the saccule is a structure called the macula sacculi. The arrangement of the hair cells in the macula sacculi cause cilia on the surface of the epithelial cells to be bent by the moving of the fluid. So if the head moves, the fluid moves. If the fluid moves, it causes a bending of the little cilia, the hairs. That's where the action potential begins. The same is true in the utricle. We have hair cells. The little hair cells have cilia. The cilia are bent by the moving of the fluid. The fluid moves if the head moves. So when the cilia are bent, then potassium channels are open, the epithelial cells depolarize, and the action potential is fired. In the saccule, we perceive motion in the vertical plane. That's up and down. If you get on an elevator, you know when it starts to go down and you know when it starts to go up. Well, that is the function of the macula sacculi. In the utricle, our little receptor there is called the macula utriculi, and that perceives motion in the horizontal plane. So you know if you're moving forward or backward, you know if you're moving left or right. The way that you know that is if the head moves, the fluid moves. If the fluid moves, it bends the cilia. If the cilia are bent, potassium channels are opened, the cells depolarize, an action potential is fired. So from the macula utriculi, we have our awareness of motion in the horizontal planes. Then we have the semicircular ducts. Notice that we have 3 of them, so we can perceive motion in angular directions. Are you spinning around is the question that we're asking here. So it's not so much are you moving up, down, left, or right. Is your head spinning around? So that is the work of the 3 semicircular ducts. The receptors located in the ampulla of each semicircular duct, those are called the crista. They also are little hair cells which have little cilia. The cilia are bent by the moving of fluid inside the ampulla so that we have an awareness of rotational motion. All right, so that's what happens in the vestibule and in the semicircular ducts. Our innervation here is the vestibular nerve. The vestibular nerve runs along with the cochlear nerve, so we just called that the vestibulocochlear nerve. It is cranial nerve number 8. The cochlear nerve goes to the cochlear nucleus of the pons. The vestibular nerve will go to the vestibular nucleus of the pons. 2 different senses. The 2 nerves just happen to travel together, so we call them the vestibulocochlear nerve. All right, I would that you simply focus on the primary vestibular pathway. It's kind of like our sense of smell. There are multiple connections made, but there is a primary pathway. So let's go then from our vestibule by way of the vestibular nerve to the vestibular nucleus. That would be the first order neuron. So that's coming from the vestibule to the vestibular nucleus of the pons, neuron number 1. Well, we are going to go from the vestibular nucleus to the thalamus. Sensory signals are processed or directed from the thalamus. Then we have our third order neuron going to the vestibular cortex. The vestibular cortex is right at the very bottom of the somasthetic cortex that we looked at that we talked about with the sense of touch. That is not shown for whatever reason, not even mentioned here on this particular chart, and I just now noticed that. That would be our primary vestibular pathway and awareness of our position in space. So you would go then from the vestibule to the vestibular nucleus to the thalamus, from the thalamus to the vestibular cortex, which is located right at the very bottom of the somasthetic cortex, the post-central gyrus of the parietal lobes. Now, all of these things listed here on this slide, these are additional connections that are made. Obviously, we are going to have to communicate with the cerebellum because the whole idea here is to know where you are in space so that you don't fall down. So the cerebellum is all about coordinating skeletal muscle contraction so that we maintain our posture and our balance. So you can take off running, but you don't fall down. Or you can step on a moving sidewalk and you maintain your balance. You don't fall down. So there are connections to the cerebellum. We also have connections to the spinal cord. That is called the vestibulospinal tract. We have to contract muscles in order to maintain our upright posture and balance, and contraction of those muscles will happen because we have a vestibulospinal tract. Okay, notice this is very strange here. The vestibular pathway has to communicate with the motor nuclei for the extraocular muscles. Well, that is the oculomotor nerve, the trochlear nerve, and the abducens nerve. Those are the nerves that innervate the muscles that move the eyeball. Well, if your head moves, your eyes have to move also. Otherwise, you would be driving down the road, and as things pass by, if the eyes don't track along with where you're going, then pretty soon you're looking backward and your car is headed forward. I wouldn't recommend that you do that. Okay, that is a very interesting thing there that we have a connection between our eyes and our vestibular sense, our sense of equilibrium. Well, the reason I mention that is this. If your inner ear sends signals to the brain that say you're moving, but your eyes send signals to the brain that says you're not moving, then you have a disagreement between the eyes and ears. That will stimulate the emetic nucleus of the medulla oblongata. We call that motion sickness. So if you're sitting in the back seat of a car and instead of looking out the window so that your eyes and ears agree that you're moving, we're going around a curve, we're going up a hill, we just crossed over a river, the eyes and ears agree. What if you're sitting in the back seat of the car reading a book? Well, you're not watching what passes by, so your inner ear is saying to you, you're moving. You're going around a curve, you're going down a hill. Well, those are signals sent from the vestibule, but your eyes are looking at the page of a book and they say, you're not moving. Then you're going to have motion sickness. So the emetic nucleus is stimulated and you get nauseated, you turn green, you might very well puke. So this is why we have connections between the vestibule and the muscles that move the eyes, so that the eyes and ears can stay in agreement about where we are and how we are moving. Well, I hope that you remember that the skin has three layers: the epidermis, the dermis, and the hypodermis. Well, situated in those three layers, we have our receptors for our sense of touch, also thermoreceptors, hot and cold, also nociceptors for sensations of pain. So some of the somesthetic receptors that are located in the skin. We have free nerve endings that respond to pain, hot, and cold. Those come very close to the surface, not into the epidermis. They are still in the dermis. The only receptors that we have that are situated in the layers of the epidermis are the Merkel disc. Those are fine discriminative touch receptors. So free nerve endings, Merkel disc, those would be most superficial. The deeper we go, the less discriminative the sense becomes. So it becomes a sense of deep pressure or vibration as you go deeper and deeper. So we have receptors called Krauss end bulbs. Those are actually located only in the mucous membranes. So they are very similar to the Meissner corpuscles. Meissner corpuscles are in the skin. Krauss end bulbs are in the mucous membranes. Notice that we have hairs, and the hair follicle itself is innervated. So if the hair moves, then we can feel it. So those are just called hair receptors. They innervate the hair follicle. If a bug is crawling on your skin and it bumps into one of your hairs, well, you can feel the bug on your skin because it moves the hairs of your skin. Okay, the Meissner corpuscle, deeper than that, getting down toward the hypodermis, the Pacinian corpuscle, and the Ruffini corpuscles. These are deep pressure and vibration sensing receptors. So these are a variety of things that give to us our somesthetic sense. So these are mechanoreceptors, thermoreceptors, and nociceptors that are situated in the integumentary system, and in some cases, the Ruffini corpuscles and Pacinian corpuscles, those are actually down deep in the hypodermis, sometimes down as far as the periosteum of the bone. So deep pressure type sensations. Now, two receptors that are not shown here because they're not associated with the skin, they're associated with muscles and tendons, and they are sensitive to stretch. So we call those Golgi tendon organs and muscle spindles. So those are somesthetic receptors that are located in tendons and muscles that perceive stretch. When they are stretched, they are at risk of being torn. So we send signals to the brain to make adjustments so that our tendons and muscles are not torn. So those are Golgi tendon organs, muscle spindles. They are also somesthetic receptors. They simply are not located in the skin. Okay, if we're talking about signals of touch, signals of pain, signals of hot and cold, and we're above the neck, well, that is the domain of the cranial nerves. So our primary pathway for those signals above the neck would be the branches of the trigeminal nerve. We have the alveolar nerves that innervate the teeth, and we have the ophthalmic nerves around the eyes. So the trigeminal nerves, left and right, have these free nerve endings that extend out to all of the tissues of the head and face. So our little pathway here then would be from those free nerve endings to the trigeminal nucleus of the pons. That's the first order neuron. From the pons to the thalamus, that's the second neuron. From the thalamus to the somesthetic cortex. So it's a 3-neuron pathway. So we are aware of hot, cold, pain, tickle, itch, touch, those types of signals coming from the head and face. Also, there are some somesthetic fibers in the vagus nerve, so our little pathway there would be to the vagus nucleus of the medulla, to the thalamus, to the somesthetic cortex. We have a very keen sense of touch around the face. So if even something as light as a fiber of dust gets on your face, around your nose, or around your mouth, well, you can feel it. You can't stand it. You have to wipe it off. You have a very keen sense of touch there around the face. Okay, signals coming from below the neck. Well, we talked about these when we talked about the pathways in the spinal cord. So we have these touch receptors, such as those that are located in the skin, or the Golgi tendon organs, or the muscle spindles. Those are receptors of touch. So from our receptor, we travel up the back alleyways of the spinal cord. Those are the gracile and cuneate fasciculi. They come together to form the medial lemniscus. The fibers of the middle medial lemniscus synapse at the thalamus. The thalamus directs those signals of touch to the somesthetic cortex, which is the post-central gyrus of the parietal lobes. Pain signals. Well, we have pain receptors. They're called nociceptors. They are going to travel by way of the spinothalamic and spinoreticcular tracts. So our pain pathway there, coming from the receptor to the spinothalamic tract in the spinal cord, that's neuron number 1, to the thalamus, that's neuron number 2, from the thalamus to the somesthetic cortex, neuron number 3. So it's a 3-neuron pathway. If it is pain that involves tissue destruction. So this is a difference between ouch and ouch followed by several cuss words. If it is pain that involves tissue destruction, then our pathway is spino-reticular. So just ordinary pain, spinothalamic pathway. If it is pain involving tissue destruction, say for instance you were chopping some wood and the axe happened to come down on your foot and chops off half of your foot. Well, you've got tissue destruction there. That's going to be ouch followed by several cuss words. So you're going to take spino-reticular pathway in that case. Signals of proprioception, well, the cerebellum is our processing area there. So we have proprioceptors, the Golgi tendon organs, the muscle spindles, those are proprioreceptors. They travel by way of the spinocerebellar tracts, and then at the level of the pons, make a 90-degree turn, go to the cerebellum. So those are our somesthetic pathways for you to enjoy. Have a good night and do well on your final exam.